Okay, today we're going to pick up on part 2 of microbial nutrition and growth, and that is for Unit 5 or Chapter 5. And so here we're picking up with culturing microorganisms. So there's different terminology that you need to know or use. You have inoculum. An inoculum is going to simply describe a sample of a microorganism. It could be obtained from 3 different sources. It could be an environmental specimen. That's where you're going to take it from a pond, a stream. It could be from soil, it could be from air. You could take it basically from anywhere, like you're growing it on a desk. That's environmental specimens. Then you could have a clinical specimen. Clinical specimens are going to be taken from patients. So from someone who's sick or from, let's say, an animal or some type of other living thing that's sick. So that would be a clinical specimen. Then you could have a stored specimen. That's where you have the inocula that is going to be grown from environmental or clinical specimens. And then you're going to take that you had it stored and you're going to use it for whatever research you're doing. For example, I order a lot of these specimens from a place called Carolina Biological, or I might order from Flynn or different types of science providers. And so those would be referred to as stored specimens. Sometimes you order them and they are living. Sometimes you order them and they're like a freeze dried, and you have to actually add in some broth, and you have to almost like bring them back and get them to start growing again. So those would be your stored specimens. Next, you have your mediums. Medium's going to be a collection of nutrients, because if you're going to grow these organisms, they've got to have something to eat. They've got to have something to live off of. So they can exist in 2 different types. You can have solid media that contain solidifying agents along with nutrients, or you could have a liquid media known as a broth. So what you see over in the Petri dish on the left, that is agar. That is going to be a solid medium. Then you have your liquid over to the side, and that liquid broth is going to be your liquid medium. Now in this, you can also have a solid and make a slant with it and grow it on the slant. And then that's how they usually actually send it in. If you order it from another company, they'll have that bacteria, if it's coming in live, they'll have it growing on the slant and they'll have the lid on it. And so it comes to you, then you take the lid off as soon as you receive it, you take it and you're going to inoculate your Petri dish over here with it and then start growing it from there. So those are 2 different ways or 2 different types of media that you can have. And next you have your culture. That's the act of cultivating microorganisms. Microorganisms that are cultivated are going to be grown on either that slant that I showed you, the one that's in the middle on the right-hand side, or it can be grown on this Petri dish that you see. Now, when you grow it, you are going to end up with a colony. The colony is going to be a culture that is visible on the surface of the solid media. So with this colony, that is usually going to be a circular shape. It could be any of these actual shapes that you see here. So try to use it without my drawing. Okay. So any of these shapes that you see here can help define or help you figure out what type of organism you're growing. So first thing you look at shape. So it could have a circular shape. It's where it's nice and circular. It could have a rhizoid shape, kind of an irregular shape. It could have a filamentous shape. Some of those filamentous ones and rhizoids are pretty cool because they look like hairs or strings growing out. So they're pretty neat. And then you can even have a spindle shape for the colony. So that's looking at the actual colony. Now with those colonies, they're going to be made up of hundreds, if not hundreds of thousands of those microorganisms, because one is microscopic. You cannot see it. So in order to see an entire colony, you have a lot of them there for you to be able to see. Another thing you're going to look at is margin. What do the edges look like? Here, you have a nice smooth, and so the entire thing's going to be the same. You can have an undulate. You could have a lobate, where you got these lobes going across. You can have what's called a curled, because you can see it kind of curling in. That one to me looks like spider webs. And then you could have the filiform, where it has the little filaments. You've got to look for elevation. And so you're pretending that you're looking at it from the side. See how this one doesn't really have anything raised at all? And so it's considered flat. You can have one that's slightly raised. We call that raised. You can have a convex, where it kind of comes up a little bit. You can have a pulvinate where it comes up even more. And then I've seen quite a few of these, and this is what you're seeing over here. It's an umbinate, and that's what you're kind of seeing over here. It's got that little dot in the center. Another one, you can have size. So when you look at the size, it could be anywhere from punctiform, or it could be small, moderate, or large. So look and pay attention to the size. Another, look at the texture. It might be rough. It might be smooth. In appearance, it could be glistening and shiny. It could be dull. It could look furry, like mold growing. Look for pigmentation. Sometimes it's cream. It could be tan. It could be white, purple, red, yellow. Sometimes the pigment's darker in the center and gets lighter as it goes out, as the ones that you're seeing in these colonies. Optical properties there. It could be opaque, it could be translucent, it might even be transparent. So you've got to look at all these different things when you're looking at those colonies. And all of that's going to help when you're describing it. That's going to help you to determine what you're looking at. Now, in order to obtain pure cultures, first you have to know what a pure culture is. A pure culture is going to be a culture that contains cells of only one species, so a single progenitor. So you want to take this and have only one type of organism growing here, which is going to be kind of hard. So what you're going to do is you're going to make this streak plate that you see here. Now, with these, to get the pure cultures, you're going to also need to know a colony forming unit, or a CFU. That's where a single cell or a group of cells can produce that colony that's there. And that's a term that's given to the progenitor, CFU, or colony forming unit. Another thing that you need to have in order for it to be a nice pure culture is sterile. Sterile is going to be a term. It means free of any microbial contaminants. So when you're starting out, things that need to be sterile are going to be your agar. You need the petri dish to be sterile, and you need your inoculating loop. Now, the inoculating loop is what you see him holding here. So it's important that that is sterile as well. There's going to be two common ways that you can isolate these cultures, and you could get a pure culture. You could do the first by doing a streak plate. That's the most common isolation technique. In this process, the inoculum is going to be spread across a solid surface of medium using a sterile inoculating loop. The petri dish is going to be the clear, flat culture dish with loose fitting lids, and it'll have some type of solid medium in it. That medium we refer to often as agar. Now, there's all different types. Lots of times you use just a plain nutrient agar. Some of them can contain blood. Some of them can contain salts, different things that microorganism will thrive off of and need. Once you streak it on the petri dish, on the agar, or that medium, then you're going to incubate it. So incubation is going to be a period in which colonies develop after adding a sample to the plate. And so we did the streaks. Notice it starts out number 1. And that's going to be your first streak that you do. And so those are the first sets of organisms. You would pass that inoculating loop then through a flame, let it cool. Then you take your second streak, you drag it across the original. And so you would do it like that. And then send it through the flame of a Bunsen burner, let it cool, and then you're going to do the same thing. You're going to drag it through this one. Put it through the flame, let it cool, and then you'll do it again. And you'll take it into this one and do this. Eventually, it gets thinner and thinner. So you will be able to isolate these different cells. So if you notice, this was number 1, very thick. Number 2, we're starting to thin out. 3, we're definitely thinning out these colonies and these microorganisms. And by the 4th area, you have nice, distinct, perfect little colonies forming there. All right, another way that you can do this would be what's referred to as a pour plate. With a pour plate, it's going to be a process in which the CFUs, or colony forming units, are going to be separated from one another using sterile or a series of dilutions. So final dilutions are going to be mixed with warm agar in the petri dish. So individual CFUs are going to start to form in and on the agar when you do this. And so you see your initial sample, you dilute it out, you keep diluting, and then you would pour. Other isolation techniques are going to be things such as broth culture media. That's going to be used for protozoan and motile unicellular algae. Micropipettes, they could be used to culture large microorganisms such as protists. The ones like Euglena or amoebas even could be used for that. Going into your culture media, we have the nutrient broth and the solid media. We looked at these before. So with that nutrient broth, that's going to be a common medium that contains a powdered beef extract and some amino acids that's dissolved in water. That's going to give the microorganisms that you put into it food and help them grow. The solid media can be made by means of adding agar to a liquid mixture. So you got your agar, you can add polysaccharides, which are sugars, things like that, and that's going to give it nutrients. That's why we often refer to it as a nutrient agar. Advantages of using the agar is going to be microorganisms cannot catabolize the agar, so they can't break the agar down, so they're going to grow on top of it, so you can see it. It also helps to withstand temperature changes, and it solidifies at the temperatures below 40 degrees or 104 degrees Fahrenheit. So when you mix it up, it's a liquid at first. So you have a liquid mixture plus agar. It's going to be poured into a petri dish, and then the petri plates are going to be made once that agar solidifies. So you would make it, let me get my thing. So what you're going to do is once you heat it up, you mix it, you're going to pour it into test tubes like this. Then you're going to send it through and sterilize these. We use a, hold on, let me exit. We put it in an autoclave in order to sterilize it. So it's like a big pressure cooker that heats up under pressure, make sure it's sterile and nice and hot. So then we take it out, we pour it in to these petri dishes right here, and then we let it set up, kind of like you would let Jell-O set up, or if you poured pudding and the pudding would set up. And so it solidifies and becomes a nice gel, almost like Jell-O is what I would say it's like. You could take a liquid mixture, you could take agar, you could pour it into a test tube even. And so if you pour it into a test tube, and let's say you want to make a slant, you're going to place that at an angle and slant those tubes, and that makes slants for you. Then you let them cool, and you have a nice sterile slant for them to grow on when you're inoculated. You're going to have 6 general culture media types. First, we have what's known as defined media. That's a medium from which the exact chemical composition is known. So all essential nutrients are going to be there. It's going to have water. It's going to have the appropriate oxygen levels, and physical conditions are going to be known. You can have what's referred to as a complex media. That's where the medium that contains a variety of nutrients and growth factors is going to be there, and that's going to support a wide variety of microorganisms. You could also have what's known as selective media. That's where the medium that contains substances that favor growth of some organisms and suppress the growth of others is going to be put in there. And so here you see the PEA pleat or the hectoen enteric agar. Those will allow certain things to grow and suppress other things. We also have what's referred to as, you see this one here, a chocolate agar or even a blood agar. And so yes, those are going to be used. We have some differential medias. This is going to be a media that's formulated to enable differentiation between different kinds of bacteria through observable changes. So it makes it so you can see different types of changes in what you're looking for, what specific type of microorganisms. We have this anaerobic media, often referred to as a reducing medium. This is going to provide conditions that are conducive to culturing anaerobes, so those that do not need oxygen. And then here we have what's called a transport medium. The medium here is going to be used by healthcare personnel, and it's going to be to move specimens safely from one location to another. Also, it's going to ensure the maintenance and abundance of that organism and prevents contamination. So they might take one of these and swab your throat to send it off and test it to see maybe if you have strep throat. Or they could use sometimes these different types of things, and they could swab the vaginal canal or the cervix and send it off and see what type of microbes might be growing there, because you might have some type of bacterial infection if you're a female. So that's how they, or even a cut, they can go in and swab it with these and send those off to the lab to be grown. Okay, special cultures that we're going to have, you can have animal and cell cultures. These are going to allow for the growth of microorganisms for which artificial media and are going to be inadequate. You can have mammal, bird eggs, living cells that are going to be used for these types of things. You can have what's known as low oxygen cultures. These are going to favor growth of microorganisms that thrive in environments that are intermediate between strictly aerobic and strictly anaerobic. So those that have oxygen and those that don't use oxygen. You can have candle jars. That's going to be where the flame consumes a lot of the oxygen or breathable oxygen and replaces it with carbon dioxide. You can have carbon dioxide incubators. That's where electronically monitored areas are going to be able to control CO2 levels and provide CO2-based environments. You've got chemical packets. These chemicals are going to be used to remove oxygen, replaced by candle jars. You can also have what's called enrichment cultures. That's your third type. That's a selective medium that's designed to increase very small numbers of chosen microbes to observable levels. And then you have what's called cold enrichment. That's going to be used to enrich cultures with cold-tolerant specimens or species. And a refrigerator is going to be used for incubation for those that like cold environments. Next, we have a way to preserve culture. So we're going to have different things that we talk about when we talk about preserving a culture, maybe keeping it, maybe you need to send it off. So first off is refrigeration. That's going to be used for short-term storage. So we're talking about days here, not very long. What this does is it slows the metabolism of cells, and it slows, because you're slowing the metabolism, you're slowing down how much they're eating that nutrient so that nutrients doesn't get exhausted quickly. The best temperature for this is going to be around 5 degrees Celsius or 41 degrees Fahrenheit. So average refrigerator. You can have deep freezing. That's going to be used for long-term storage. That could be years. Like I said, some of the specimens I order are freeze-dried. The best temperature is going to be around negative 50 degrees Celsius to 95 degrees, or that's negative 50. It could be negative 58 to 139. Negative 139 Celsius. So any of those, depending on what lab it's coming from. And when you get it, you put it in your freezer and you keep it there until you need to re-inoculate it with some broth and start growing it again. Then you have the third type, the lyophilization. This is going to be used for long-term storage such as decades, so very long. That's going to involve the removal of water from a frozen culture within a vacuum, and so nothing else is there. So those are going to be different ways you can store. Now, microbial population growth usually goes by very quickly. Like E. coli, it divides from 1 cell to 2 every 20 minutes. The average, every 20 to every 30 minutes. So this one here is showing you a diagram that's using about every 30 minutes. So what is binary fission? Well, it's going to be a process in which a cell grows to twice its normal size and then divides in half to produce 2 daughter cells of equal size. So it makes itself bigger, it starts to replicate everything from within, and then it starts to divide. That's what you see up there. This process is going to be how most of these microorganisms divide, how they reproduce. Now looking here, we have our mathematical considerations. We have what's referred to as exponential growth. That's going to be an increase in microbial population size dramatically. So cells here are going to double in a fixed interval of time. So 2, then 4, then 8, then 16, and so on. Generation time. is going to be defined as a time required for bacterial cells to grow and divide, or the time required for a population of cells to double in number. Most bacteria are going to have a generational time of 1 to 3 hours. And that's going to be dependent upon chemical and physical conditions. Are they all there? Is everything being met? Do they have what they need? Now, with these population growth phases, you're going to have specific things. You're going to have what's called a growth curve. That's going to be a graph that plots the number of bacteria growing in a population over time. Bacteria grown in broth is going to have 4 distinct phases of population growth. So with these population growths, first you have what's called the lag phase. No significant increase in number of cells. You're going to have cells that are adjusting to a new environment, and they're metabolically pretty much inactive. After the lag phase, we go into what's known as the log phase. Here we have significant increase in the number of cells. The cell numbers are going to be increasing exponentially, and they are metabolically active. So we have now metabolically active cells that are eating and growing and reproducing. Third, we're going to have what's referred to as the stationary phase. No overall increase in cell number. Cells start dying and equal to new cells forming, so birth and death rate basically equals out. Cells dying due to lack of nutrients and buildup of toxic metabolic waste products are going to be common. So that's what's they're running out of food, waste is building up, and so we're on the down phase now, that stationary phase. And then we hit the fourth phase, which is the death phase. Exponential decrease in the number. Cells die faster than new ones are formed. We have a lack of nutrients because the cells from the previous phases have used up all that nutrients, and now a lot of these cells are even being poisoned by their own metabolic waste products because of the buildup of waste there. And so here you're going to be able to see the lag phase, no real growth, not metabolically active. Hit the log phase, exponential growth. They're active metabolically, they're growing, they're dividing, the numbers exponential. Stationary phase, we have equal growth and death. And then death phase, where they're starting to die out, they've used up their nutrients, and they're starting to be poisoned by their own waste. Now, here we have continuous cultures in what's referred to as a chemostat. The chemostat is going to be a device that's used in research and industry to maintain microbial cultures in a particular phase. So it's going to involve a supply of fresh medium while removing spent medium. So it takes away the medium that the nutrients has been used up in. 2 main uses for this: to study microbial population growth under specific conditions, and to maintain constant production of industrially useful microbial products. So that's why they would use this. So measuring microbial reproduction, you could have direct methods like serial dilution. So here we have a stepwise dilution of liquid culture in which dilution factors at each step is going to be constant. This is going to involve a viable plate count. So with that, there's going to be an estimation of size of microbial populations based upon the number of colonies formed when plated after serial dilution. This is going to underestimate the number of cells present in the sample, though. So you've got to watch this type. You could have what's referred to as a membrane filtration. This is going to take a large sample that's poured through a filter small enough to trap cells. The membrane is then going to be transferred to a solid medium and then incubated. Here, you're going to have colonies that are then going to be counted after that incubation. Most probable number. This is going to be a statistical estimation of microbial population size that's going to be based upon sample dilution that's required to eliminate microbial growth. And so it reduces the number down to 0. You can have a microscopic count. The sample is going to be placed on a cell counter. The glass slide with basically an etched grid on it is going to be there. It's going to be viewed through a microscope. This involves counting the number of bacteria in several of the large squares, and then you calculate the mean number of the bacteria per square that you see on there. This doesn't require incubation to do this one. You can also have an electronic counter. That device is going to count the cells as they interrupt the electrical current flowing across a narrow tube that's going to be held in front of the electronic detector. And so that's going to be useful if you're counting large numbers of cells or large cells. There's some indirect methods. You can have what's called a metabolic activity that estimates the number of cells in a culture because it measures the changes of nutrients being utilized and waste products or pH that's being produced. You can do a dry weight. That involves filtering, drying, and weighing the organisms. And that's going to be suitable for broth cultures because it's hard to see with a broth. You can't see colonies. It just gets foggy looking. So over to the left, if you're looking in the middle, it's nice and clear. If you look at the one beside it, it starts to get foggy. Turbidity. That involves measuring changes in the turbidity or cloudiness. That's what I just discussed with the broth culture. You can have a spectrophotometer, and that's going to measure the amount of light that's transmitted through the culture. Transmission is going to equal inversely proportional to the size of that population. So the larger the population, the less light is going to be able to go through, and it won't reach the detector. And more light is going to be absorbed that way. This does not distinguish between living and dead cells, though. So that you'll have a buildup of living, a buildup of dead, and you won't be able to know how many living are actually in there. You can also do what's known as a genetic method. That's going to involve isolating unique DNA sequences representing different kinds of prokaryotic species. So that's going to end us for Unit 5, and we will meet back with Unit 6 and talk about controlling microbial growth. See you then.